Thank you very much for being here, and Michael, thank you for inviting me, but even more, thank you for proposing this title for my talk. I have never talked about life in a scientific talk, but it's very interesting. And as I was preparing it, I thought that actually, over the time when I was working on what I'm going to talk to you about, I changed my interest in science, and my life changed quite a bit. So I could see it as something personal. So thank you very much. So I started as a chemist, but by the time I retired, I was really a cell biologist. And so I'm going to talk about microtubules, which are important components of all our cells, practically. And in particular, I'm going to talk about microtubules in skeletal muscle, because that's what I did research on for the past probably, I'm not sure I want to say how many years, but at least 30, probably a little more than that. So, and I'm going to focus on the organization of the microtubules, which actually look quite beautiful, I think. I hope that we will agree on that. And I will start with my first image. It's a little bit saturated, but I don't know if it's, I think that the image is better on that screen. It's less saturated, but never mind. So what this is, sorry, not what I'm sorry, I'm going to, I would like to go back.
Press the left button.
Yes, I did, but yeah. Okay, I'm sorry. Sorry for that. This photo was taken. Sometimes I will put the name of who took the image. In this case, it was a friend and colleague of mine. And this is an image from a cell that's actually a human cancer cell. So not something very cheerful, but anyway. And what you see in green are actually microtubules. The name would suggest, small tubes. You will see that there are indeed tubes in the cell. And what you see in blue is the nucleus of the cell. Each of our cells has a nucleus which contains our genetic material, all the DNA. The way we obtain such images, I call them micrographs because they are taken on a microscope. They are photos that you take through a microscope. So the cell can look enormous, but it's actually a fraction of a millimeter, or of a, I prefer to use the decimal measures, but anyway, a cell is a very tiny thing. So you see these tubules are concentrated. They originate somewhere near the nucleus. I will tell you how they do that, and they go towards the limits, the membrane of the cell. They have many different roles. They are important when the cell divides into two daughter cells. The microtubules serve to carry the genes and put them together in the daughter nuclei, let's say. So they're very important for cell division. They are also important for strengthening the cell. A cell often moves, and when we talk about skeletal muscle cells, they do move. If I contract my muscles, the cells really are subject to some forces, and it's very important to have something like these microtubules to help keep their shape and their strength. So I'm now going to show you a completely different type of cell that has a very different organization of microtubules. So here, instead of this shaggy hair that we had in the previous image, you can see, I don't think that, you can see these longitudinal lines and transverse lines. The microtubules here look really like a regular grid. And the first time I saw that, I was really stumped, and I decided I had to understand how this is organized. Skeletal muscle is about the only type of cell that has this kind of organization of the microtubules. And also something that I will come back to that, in my I'm sorry. In skeletal muscle, one cell has many nuclei. That is a change that occurs during development. I'll show you some cartoons explaining how this takes place. Since we're talking about skeletal muscle, I just wanted to show you one image of someone who was working and lived very close to where I was born, because Vesalius was working in Flanders and I was in Brussels, which is very close to where he used to be. Anyway, a long time ago. And he was one of the first ones to dissect entire cadavers. And if you can see here an arm that is indicated with the red arrow, and you can see I hope see the thin lines that divide, the cells form groups. So the skeleton, the cells of this arm are a bit smaller than what we can than what we can distinguish, but they form cylinders, and that's what he started studying that, but he could not see them under potent microscope and to see the individual cells yet. This is another cell from a muscle, and you see again in green the microtubules and in blue the nuclei. But in this case, you can see that the microtubules are very disordered. This is because this is a mouse muscle cell, and it was part of a work where the muscle was injected with a toxin that really damages the muscle, but the muscle then regenerates. And what you see here is a stage of regeneration where there are microtubules, but they're very disordered. And I will come back to notion of regeneration, degeneration in the muscle. So now I want to explain to you a few words of what these microtubules, what they're made of, because you see some green lines that doesn't tell you anything about the chemical or cellular composition of these microtubules. So they consist of strands of proteins. You know there's protein in meat. I mean, I don't know if you know what proteins really look like let's say in a textbook, but anyway. So they are, okay, this is where I was. They are cylinders made of strands of proteins, and each of the colored blobs represents one molecule of protein, and the protein that makes the microtubules is called tubulin. It kind of makes sense. There are 13 strands that form these little cylinders, and they're dynamic. They're constantly growing and shrinking, and that allows them both to strengthen the cell, but also to transport other proteins and to transport nucleic acids through the cell. So they really have quite a number of roles in the function of the cell. Now, the tubulin, the protein that makes these microtubules, is very diverse. You see here that let's say the blue blobs represent an alpha tubulin, and the green blobs represent a beta tubulin. That may not say very much, but just I want to emphasize that there are 7 to 10 different genes for alpha tubulins and 7 to 10 different genes for beta tubulins, which means that you can have really a lot of different microtubules in a way, and in some cases. That's true. That's true. But it's not. It's not a closed. It's not a really closed loop. It's like an assembly of these 13. And don't ask me why there are 13. I really can't tell you. Yeah, that's right. It's life at different levels. That's 13 here. So you can, you imagine that with so many different forms of these tubulins, you can have microtubules that have different chemical or biochemical properties, and some of them may not be totally healthy. We'll come back. Yes. Well, very often proteins can hold together because you may have some parts of the protein that is charged positively and some part is charged negatively. But if they're surrounded with water, then the charge is less important, but there may be some parts that like to stay together because they are not charged and they're like fat a little bit, and so you have different ways to keep them together.
It's electrostatic forces that hold them together?
Electrostatic forces can hold them together, except when if they're in a medium like in water, it's going to reduce the strength of the electrostatic forces. Yes. So now what I want to show you is how the microtubules are put together, how they're what's called nucleated. And so I talked to you about the development of muscle from a precursor cell, or what's called sometimes a stem cell, and what's called actually a myoblast. If you get to a scientific journal, even if it's not very specialized, if you see myo, it has nothing to do with a condiment, but in general it has something to do with muscle. And blast is often a precursor cell. So myoblast is a small cell which is the precursor of muscle, and muscle does not continue to divide when it is mature, let's say, but you always have stem cells that are going to be able to regenerate and to help the muscle regenerate, which is important for us if we if we damage our muscles. So I can't really point to the cell to the right, but in blue you see the nucleus, and you see the green shaggy microtubules seem to originate from this little yellow dot. This yellow thing is called the centrosome. Although it is rather small in a cell, it contains at least 200 different proteins, including one that's a gamma tubulin. So you have alpha tubulins, beta tubulins, gamma tubulins, but I'll stop there. I don't want to make a whole alphabet soup. And there's something in red that's an organelle that's important for transport of proteins in the cells, but I will not get more into it. But just you will see that it changes during development. So this is the way we get, these shaggy microtubules. They originate in this small assembly of proteins, the centrosome. But at some point, let's say if we're in a lab and you have a dish with these precursor cells, at some point they're going to get very dense, and that's going to start them. They're going to start differentiate, which means that they're going to start to be different. They're going to acquire muscle characteristics. And what's happening then is that two cells are going to come together. The membrane of the cell is going to open, and you will have then a compartment with two nuclei. And then a third cell may join them. You will have more, and they will elongate. And that's how you will get what we call a myotube, which is a multinucleated cell, and which is elongated. And what you can see from the right is that the microtubules, the centrosomes, and the organelle in red are all changing dramatically. The microtubules now become all elongated. They follow the axis, let's say, of the muscle. The centrosome seems to disappear, but its components now form a rim around the nucleus. And this red organelle has now divided itself in many parts that are also kind of more or less elongated along the muscle, the forming muscle. And then we get to the, let's say, not exactly the final, but the next stage, which is really much more close to a mature muscle, called muscle fiber. And you see these striations, these black and white lines, represent actually the myosin and actin, which are the muscle proteins that are going to contract when we want to move our muscles. And they are contracting because they get a signal from a nerve. And the little red squiggle that's touching that muscle fiber represents actually a motor nerve. That's a nerve that comes from far away from the brain and goes to the muscle and gives it, some electrical jolt that's going to cause the proteins to contract and your muscle to move. What happens to the microtubules during that stage is that they start forming, if they're the right type of muscle fiber, they form the grids that I have shown you earlier. The centrosomal proteins are, they seem to aggregate at the poles of the nuclei, and the little red compartment is divided into much smaller parts. And what's interesting, sorry, yes.
I'm a little confused about how you get 13 these tubes from this structure. How do they form together in tubes? How do the tubes come together? Is this the formation of a tube?
Well, this is very small compared to the whole tube that I was showing you was really one microtubule. So indeed, that would be the one grid. Let's see. Sorry, but when we think of the muscle fiber, it is more than one of the components of the tube. I could make you a little cartoon or something like that.
The green lines there are the microtubules, correct? The green lines are the microtubules.
Yes, the green lines are the microtubules. So yes, so actually right, the green lines, each green line has these assemblies of tubulin. Yes, yes. So, but now all I have told you about is about something that happens in healthy development, in normal development, and in healthy cells. But unfortunately, they are diseases. They are muscle diseases, and one of these diseases, which is really very, very damaging, is Duchenne. It's called Duchenne Muscular Dystrophy. It was discovered long ago. The gene that's missing in the people with Duchenne Muscular Dystrophy is called dystrophin, and it was characterized about 40 years ago. One knows the sequence of the protein, one knows the sequence of the nucleic acids that make this gene. I can't say that there's no cure at all. There are many labs that are working on cures. There are animal models. There's a mouse model for the disease. And actually, what you see here is the upper image is from a mouse, from a normal controlled mouse. But the lower one is a muscle fiber from a mouse, the name of which is MDX. Nothing to do with the Acura MDX. I don't know why they use that. But MDX is just, it's a mouse model for Duchenne muscular dystrophy. So it lacks the gene that is responsible also for that disease. And what you can see is that, again, the microtubules, as we have seen, for example, in the image of a muscle that was regenerating, you can see that, again, the microtubules are not completely normal. They're elongated, they go in all directions, they do not form the regular grid.
Evelyn, I have a question about these images. Are they optical photographs and are the colors from dyes, or how are they green and blue?
Yes, yes. Thank you. I was going to mention in the beginning, and I did not. So yeah, this is called images of immunofluorescence, because what you use is that, so we have, let's say, cells growing in a dish. You can fix them, you can permeabilize them, and you can add reagents, for example, antibodies that are against tubulin. So they're going to bind all the tubulins on your dish, and if you can get fluorescent antibodies. They have a fluorescent tag. It can be green fluorescence, it can be red, as you like. You will then fix the whole thing. Yes.
Are those two pictures? Are those those photographs of slides or tissue, or how? Or are they electron microscope, or where do they come from?
No, I mean this is light microscopy, not electron microscopy. Although we have done a lot of electron microscopy as well.
It's optical.
But this is optical.
Oh,
yes.
okay, yeah.
This is optical microscopy. And for example, for the nuclei, there is a dye. The name doesn't matter too much. That dye binds the nucleic acids and fluoresces when it binds them. So that's how you can see the different parts of a cell. And actually, the color can be real color or can be not.
Oh, that's a lot bigger than one of those tubes.
So this is what you see. That is but it is one cell, and you can. It's interesting. You can dissect. You can take a mouse muscle, and with tweezers you can dissect and get single fibers. And these are single fibers because they are easier to label, to permeabilize, and to label. So one thing is that I will stop my voice. This is. Oh, thank you very much. So this is just the title of a paper that was published by some colleagues of ours from the medical school at the University of Maryland in Baltimore. And they're also interested in microtubules in muscle and in this disease. And they did a number of experiments from which they concluded that the microtubules were not, the defects in microtubules in this disease is not just a consequence of the disease, but it also takes part, and it's the microtubule damage actually is part of the pathology of the muscle. So that was very interesting. That was a very interesting news, let's say, for us to learn, because some people could say, oh yes, okay, the microtubules are different, but what does it do? That's not the important part of the disease. But it looks like it may be the important part. Okay, but now we're going to get to a little bit of molecular biology, because what this group did and what we did also is to do what's called RNA sequencing. You can, for example, take a certain cell and you can sequence all the genes or all the DNA or all the RNA and see what defects there are in the genetic composition of the cell. So they got samples from a certain number of children with Duchenne Muscular Dystrophy, and also from controls, from normal people without such a disease, and they were able to sequence all the RNAs in those. And later we did that on mouse muscle. And what they found, they published all the differences. So in this case, this is just a small part of a figure that they have in their publication, where they show just by coloring. I don't know if you can see the names. So here, this is they show microtubules, and then just on the left, all the names just on the left of the word micro are all the alpha tubulins, and on the right of the word tubule are all the names of the different human beta tubulins. Red shows genes or RNAs that are increased in the disease. Blue shows some that are decreased, and gray shows some that are not changed. So the alpha tubulins are not changed that much, but the beta tubulins, you can see that most of them are increased. And I have put an arrow towards the first one, which is top 6. Because I had read about top 6, it has some features that I thought were very interesting in this context. And the features of top 6 is that someone who's also retired, Dr. Cabral at Baylor College in Houston, did a lot of studies of the individual tubulins and to see what their roles can be, if they're, and top 6 was very interesting because he found that it's a minor tubulin. It's only 10% of the tubulins at most, generally in most most but it is ubiquitous. You don't see, if a cell has microtubules, it will have some of it. But the second line is more important. He said that it's essential but toxic when overexpressed. So if you have more, if instead of having just 10%, you have 20%, 30% of your tubulins are top 6, that may be bad for the microtubules. And what our colleagues and we found also is that in all the models of this disease, whether it's the mouse or in their case they had not the model but real samples, top 6 was very much increased. It was increased 3-fold, 5-fold. So if really that protein is toxic, if that tubulin is toxic, we thought that that might be a reason why these microtubules in this disease were disordered. So we started working on that. We hypothesized that perhaps top 6 had something to do with the defects and with the pathology of that disease, and I very much wanted to investigate that further. So here, I can't truly, no, okay. These pointers do not work on reflective screens, but anyway, what you see here now is a section of a muscle of a mouse, and this is not the. Sometimes we can make very thin sections for electron microscopy, for example, but these are fairly thick sections for optical microscopy. And again, these muscles have been stained in red, or okay, what appears in red is top 6. What appears in green is not actually the microtubules. Let's say here, the only microtubules that we could see would be from those that contain top 6. And just what I would like to show you on the left side, you have on the left column you have the wild type, the normal muscle, and on the right side you have this MDX muscle, and you can see that there is a lot more of these red muscle fibers on the right side in the MDX, and so they contain a lot of these top 6. Also, I don't know if you can see that, but the top 6 fibers appear to have the nucleus in the center, and that's also a sign of abnormality, but also of regeneration. I did not emphasize that, but when I had the cartoons showing the development of muscle, in the myotubes, the nuclei were in the center of this tube. But when the cell matures, the nuclei are just under the membrane there, at the outer part of a muscle fiber. During regeneration, they're in the center. And so you can see that in this muscle, you have a lot of cells that overexpress top 6, and they're regenerating. So that was an interesting, I was very interested in finding that. Davide Randazzo, who was a postdoc working with me and who's now replacing me at the NIH, did different experiments. What he did was to overexpress top 6 in mouse muscles. So you can inject what's called a plasmid. It's a piece of DNA or DNA sometimes with a fluorescent tag, and it will be expressed where you inject it, and then you can see what things look like. And again, you can see the top part shows again an increase in microtubules, but they are very disordered, and that's when you increase this top 6. The other panels are controls, and they have normal microtubules. So now, this is the last I wanted to tell you about top 6 and Duchenne Muscular Dystrophy, but I just wanted to mention a very recent study, a very recent paper. This was published in April 2024, and the title may be difficult to understand, but this is a study of genes that are different in people who are left-handed. So it was a study of handedness. 90% of people are right-handed, about 10% are left-handed. The study was done by a group in the Netherlands, and they used data from a biobank in England. And I just thought I would take this slide that was done at the NIH explaining what the technique that they use is. This is what's called this GWAS, Genome-Wide Association Studies, and it is done a lot if you want to understand what genes are maybe associated with a disease or with a difference. I don't know if I would not call being left-handed a disease, but what is associated with that. And just the numbers, they had 38,000 in the bank of data that they could use. There were 38,000 left-handed people and 313,000 right-handed. So it kind of represents more or less the percentage of left-handed to right-handed people. And what they discovered is that there was one gene that was very well associated with left-handedness, and this is top4. So it's not top 6, it's top4, but it's another component of the microtubules. And in the summary of the paper, it said, the study reveals a role for rare protein-altering variants in left-handedness, providing further evidence for the involvement of microtubules and disorder-relevant genes. So anyway, I was very surprised reading that you know, a tubulin has something to do with being left-handed. Anyway. And finally, this is just, I really always wanted to be able to get some human muscle samples to see if in a normal muscle we have these grids. And finally, I was able to get it through talking close with a colleague and friend and collaborator at the University of Copenhagen. And so this is a staining that I did on a sample he sent. And you can see that the microtubules shown here in red are perfectly not, you know, these were samples that had been kept fixated for a long time. But anyway, you do find a regular grids in normal human muscles. So anyway, DMD human muscle is not available for research, but it is good to know that the healthy muscle is a good model for, you know, finding out what happens in the microtubules in diseases. And okay, so that's it. I hope I was not too long. Just microtubules are important for cellular organization. In muscle, microtubule defects are implicated in Duchenne Muscular Dystrophy. In brain, microtubule mutations are involved in left-handedness, and it is critical to continue and make progress in understanding how microtubules are formed, regulated, and what role they play in health of muscle and other tissues. Thank you.
Thank you very much. So we have time for questions. I'll pass the microphone to you.
Are microtubules the same as nanotubes?
No, there's a factor of 1,000 between nano and micro, and so no. Nanotubes are generally something that is fabricated, that is made. Yeah, so it's not the same.
Nanotubes are just carbon atoms in the tube. Yeah. I have a question about neurons. You talked about muscle cells, but neurons are other cells, and the same things apply there.
Well, neurons have, neurons are important for the whole thing, but they are very different, and their organization is very different. Although neurons contain a lot of microtubules, you know, neurons have the cell body where you have the nucleus, and then they have many long, let's say, I wouldn't say filaments, but they extend arms that are going to contact, for example, to muscle and to other organs that need to be innervated. In neurons, for example, in general, the microtubules are just elongated along these processes.
So you showed the microtubule, the center, are there fluids flowing down the center, or what goes inside the tubes, microtubules?
I don't think there are other proteins in the center. I think that because there are some proteins that bind microtubules, and some of them have a binding site for tubulin that is in the center, in the opening. So I don't imagine that there's a lot of transport of liquids, although, of course, you know, we would have the opening is in the order of 10 nanometers, something like that. So it's rather small. But I can't exclude. I mean, you know, there's enough space for certainly water or
maybe water. Yeah. Other questions? Please wait for the microphone. Please wait for the microphone. Wait for the microphone.
Do the microtubes join together and molecularly and form the long tubes, or how are the longer tubes formed?
They don't truly join and then separate, but they keep growing. They keep being, they keep kind of forming. They are nucleated and they keep growing. But at some point, something may happen, and then there are images showing the strands of the tubes kind of opening, and then they start decreasing very fast. And then they can be also, there are some modifications of the proteins in the tubes that are going to stabilize them. So yeah, I didn't want to go into all the all the details.
Okay, David.
Since it's been some hundreds of years since we started to understand the higher level structure of muscle, when were microtubules really described and began to be studied, and when did it start to be understood how they're organized?
The study of microtubules, I would say, has progressed together with the progress of microscopy and of techniques, because the more, the microscopes that we have now, the optical microscopes, reach a resolution that is similar to that, or nearly similar to that, of electron microscopes. So we have, I don't know if you heard, the Nobel Prize a number of years ago was given to a group of people who developed super resolution microscopy. It means that, for example, in some of the images that I showed you, you would see one green line and you might think that it is one microtubule, but it is not. When you can increase the resolution, you will see that there are actually several microtubules there. And in the past, you would have needed electron microscopy, which is a lot more difficult to carry out. But now there are techniques of optical microscopy that's allowed to do that. And so I would say that took place in the 1950s. There were a lot of development for using electron microscopy for cell biology, but super resolution was something that took place in the 1980s, 90s. So it's more recent. It's more recent than Vesalius.
I'm not sure if this is a sensible question, but if there's a degradation in the tubules in Duchenne's, where the grid is sort of lost, I guess, is it possible that in a normal human being, like exercise could increase the formation of grids in your muscles?
Well, I don't know how you increase the formation of the grid, but certainly exercise. Sometimes people will have some local damage in their muscles when they exercise a bit too much, perhaps. So I would imagine that you have some patches of the microtubules which are not really in perfect shape. But I think that there will be a regeneration, and it will be okay. Yes. But there are other diseases where the microtubules are not normal. They're a bit like the canary in the coal mine. If you see microtubules that are not normal, it suggests that the muscle has a problem.
What other diseases would be in that same category?
So the question was, what other diseases are in that category? So, for example, a disease which I collaborated with Nina Reben, a scientist who's not at NIH anymore, but still working on that. And she's been working for a long time on something that's called Pompe disease, which is also a very pretty awful disease. This is a disease of the the lysosomes. Lysosomes are little organelles that we have, for example, in muscles, and where when you have, lysosomes are like the garbage bags of the cell. But in the disease, they do not work, they do not function. So when a protein needs to be discarded from the cell, it doesn't work, and there's a big accumulation of very large patches of just, you would say, kind of garbage. And that prevents then the muscle from contracting normally. And indeed, the microtubules are also abnormal in this disease. So, yeah. And sorry, I think that there are also all of the microtubules and the organelles that are kind of interacting with them also have problems in Lou Gehrig's disease, which is more that, but that's more in the brain. That's more in the brain than in the muscle. But
I've always heard that weightlifting tears muscle. Weightlifters, bodybuilders tear the muscle in order to make it grow more. How does that work with the microtubules? And does all exercise tear muscles?
That's why I don't do lifting.
Get back on the couch.
I don't think that I have a perfectly detailed answer to your question. Yes, you can tear muscle, but it will, in general, muscle that's torn will be repaired. And I don't know exactly, we have not had a chance to look at the microtubules of torn muscle, especially of human things. So,
is there one more question? You have one more question. Hey, please wait. Wait. So,
I didn't hear right, but it sounded to me as if you were saying that the optical microscopes today are near the resolution of electron microscopes. I'm interested in the equipment. If you didn't have the equipment, you couldn't do all this.
Yes, it's true. I mean, what you can do is very much dependent on the equipment. I mean, there are still, electron microscopy remains interesting and important, but both techniques, when you push them very much, are not so simple. when you want to work at super resolution optical microscopy, you need to have, I don't know how many lasers on your microscope, and it is rather expensive and not necessarily rapid. It's not you take your slide, you put it under the microscope, you turn on, you switch on the light, and you say, oh yeah, beautiful, thank you. So it is a lot of work, but it is fascinating. And actually, my job, part of my job at NIH, I was the head of what's called a light imaging facility, because this equipment is very expensive. So you can't have every lab, even in a place like NIH that has good budgets, you cannot have every lab having the same type of microscope. And it would be an excess because it wouldn't be used all the time. So for each institute, you have a group that has, concentrates all the microscopes that the institute can afford. And then, so that's what we were doing. And therefore, we would then have postdocs or students would come to us. We would teach them how to use the equipment. We would also advise them about what to do for the questions they were interested in. And that was, that was very interesting.
The resolution depends on the wavelength, right? So if you push the wavelength towards ultraviolet, extreme ultraviolet, X-rays, then.
Yes.
So, but your work is with visible light.
Yes. Yes.
But are there similar projects with ultraviolet light or even?
Well, close to it. The dye that binds to DNA and that we use for the nucleus is in the blue, fluoresces in the blue, and in the.
Any more questions? No, thanks, Evelyn, again.
Thank you very much.